my pleasure to introduce Dr. Anor. Dr. Anor is a serial chemist in the Department of Food Science and Nutrition at the University of Minnesota. His expertise is in carbohydrate chemistry, and he's been involved in end-use characteristics of wheat in collaboration with the University of Minnesota Wheat Breeding Program. He's responsible for preparing pasta, regular and sourdough bread, and analyzing the samples for their glycemic index. He's also responsible for analyzing wheat samples for the amylose, resistant starch, FODMAPs, fructans, and amylose trypsin inhibitor. So we welcome Dr. Anor. In addition, we're very pleased to have Dr. Anderson. Dr. Anderson leads plant improvement programs focusing on wheat, intermediate wheatgrass, also known as kernza, field pennycrest, and research on each species and includes cultivar development and the investigation of breeding and genetic principles related to crop improvement. Research is focused on identifying genes underlying economically important traits and implementing marker-assisted and genomic selection for cultivar and germplasm development. He will oversee and direct the field and lab technicians and screen for high amylose, low and high ATI and FODMAP populations to grow in selected lines, produce grains for analysis by Dr. Anor. So with that, I will turn things over to our guests today. So welcome, Dr. Anderson and Dr. Anor.
Thank you very much. I guess I would start the presentation, so I'll share my screen, and hopefully you guys can see the presentation. Okay, so thank you very much. I'm very happy to be here today to share some results of the research we've been working on. Of course, it's myself, Jim Anderson or James Anderson, and then Praveen, who works with Jim. So today we'll be talking about tackling wheat digestibility, FODMAPs, and ATI levels in wheat lines. So why did we do this work? So it was all based on the fact that the per capita wheat consumption in the US is actually declining. And the decline is so worrisome such that if you look at data from 2000, this is just recent, 2000 up until 2018, you can see we have a very steep decline in the consumption of wheat. Now, this steep decline, of course, would inevitably affect the income of farmers and also affect the industry involved in the processing of wheat. Now, why this decline? It could be due to a couple of factors. Some of the factors that we have identified includes what we call fat diets. So there have been the promotion of these diets that has resulted in a huge population, a huge number of people actually removing starches from their diet. And if you look at wheat or cereals for that matter, it's basically starch-based. So if people are removing starches from their diet, it also results in a reduction in the consumption of wheat. Some of these diets are what we call paleo diet, where people are eliminating gluten, which is a problem with wheat. And then we have what we call Whole30 diet, where people will only eat veggies, fruits, and nuts for 30 days. And then we have the ketogenic diet, where people are looking at high fat, low carb keto diet. So all these diets that are being promoted now is really hurting the consumption of wheat. And of course, in addition to that, we know people are avoiding gluten because of celiac disease. And this is a huge problem, even though it's just a small percentage of our population that is affected by gluten. It's become a huge thing, such that people who don't even know they have celiac disease are avoiding wheat altogether. So when we come to the avoidance of gluten and wheat, we know the problem is from gluten. Gluten is found in wheat, rye, and barley, and it results in celiac disease, which most of us know as an immune disease, where people just cannot eat gluten because it damages their small intestines. As I said earlier on, just less than 1% of Americans have celiac disease. But again, it's been a huge thing out there. The other reason is also wheat allergy, right? So wheat allergy is when people just react to wheat proteins, not necessarily gluten, but they react to wheat proteins when they eat them. that has also resulted in people avoiding wheat altogether. Then the third one is what we call non-allergy, non-celiac wheat sensitivity. So this is where people react to wheat, but we know it is not from celiac disease, and we know it is not from wheat allergy, right? So everything is just going against wheat, and we thought there is the need to do something. So let's look at the NCWS. So if it is not gluten, if it is not wheat allergy, then what is really the problem? Now we think it is from what we call FODMAPs, right? So these are fermentable oligo, di, and monosaccharides and polyols. It's a very long acronym, but we kind of cut it short as FODMAPs. These are mainly small sugars, monosaccharides, or disaccharides. So they mainly fructose, lactose, or what we call fructo and galacto oligosaccharides. Um, and then we also have polyols, such as what we call, these are sugar alcohols, sorbitol, xylitol, and mannitol. Some of these sugar alcohols are actually used as in low calorie diet. So most, if you buy a gum that says low calorie diet, you basically might have some of these sugar alcohols. And most of you probably might have experienced that if you eat a lot of these sugar alcohols, or if you eat gum that has a lot of these sugar alcohols, sometimes it gives you a little bit of laxative effect. And so when people eat wheat or eat foods and they have that laxative effect, it becomes very uncomfortable, so they will avoid it. The other reason is what we call amylase trypsin inhibitors. These are small molecular weight proteins in wheat. They are not gluten proteins, but they also result in also actually result in irritating the intestinal wall, resulting in a lot of discomfort to consumers. So we have a problem that has to be solved. Now, if you look at this FODMAPs, right, that I just mentioned, FODMAPs can be present in a lot of other food products, right? So if you look here on this slide, you see it's available in asparagus, in onions, in garlic, in a lot of other products that we consume every day. Now, it has been estimated that if you have less than 3% or 3 grams per 100 gram of 0.3 gram per 100 gram of FODMAPs in your food, people can actually tolerate the discomfort that it actually generates. Anything more than that, it becomes problematic. So if we are looking at this, you can see that if you come to this graph here, you can see most of the cereals that we've been consuming, brown rice, white rice, basically don't have anything. Pasta from quinoa, pasta from gluten-free product, basically have ATI, FODMAP levels that can be tolerated. But if you look at pasta from wheat, right, whole wheat bread, a biscuit from cereals, couscous from wheat, you can see they are really all above the 0.3 cutoff that we can tolerate. So it means that the wheat we consume every day probably may be contributing to some of this discomfort. So we decided to tackle this, and this wasn't going to be done from the University of Minnesota alone or from ARI. We put together a team from Minnesota Wheat and Research Council, of course, the University of Minnesota, and from ARI, and from Back When Foods. This is a private food company owned by Brian. We decided to tackle this issue, and therefore we put a proposal together, submitted it to the Minnesota Department of Agriculture, and it was funded. So that was the beginning of this project. So what were the long-term goals of this project? Our aim was to try to reduce the discomfort that results when people consume wheat-based products, right? When we do that, we intend to also improve the health of consumers, because then they won't have to have all these problems from the consumption of wheat, and ultimately results in the profitability of wheat farmers, because if we are able to reduce the discomfort, we anticipate there will be an increase in demand of the consumption of wheat, resulting in farmers making some money. So the specific aims of this project is to identify wheat varieties that naturally would have low anti-nutrient levels. When we say low anti-nutrient levels here, we are talking about the ATIs and then the FODMAPs, right? So we want to identify wheat varieties that probably may have very low amount of these, and then be able to see whether we can have any genetic link. So the breeders like Jim Anderson would be able to breed for wheat varieties that have a low ATI and FODMAPs. In addition to that, we want to also see whether we can use techniques like fermentation to actually reduce these FODMAPs. Now, these FODMAPs, remember, are sugars and therefore can be fermented by microorganisms. So we hope that when we ferment our foods, the microorganisms that are involved in the fermentation will be able to use up these FODMAPs, therefore resulting in products that have low amounts. So the specific objectives would be to characterize variations and identify genetic markers for both FODMAPs and ATI in ancient, heritage, and modern wheat varieties, right? Jim probably would explain that more in detail. But not only are we looking at these ancient, heritage, and modern varieties, we want to look at these varieties from different locations. So we specifically worked with St. Paul and also Crookston. We want to see whether the locations have any effect on how much of these FODMAPs and ATIs are in this wheat samples. And of course, use the fermentation to be able to reduce the FODMAPs. For the fermentation piece, we'll be working with Brian of Backward Foods. He will be doing all the fermentation, and then we will do the analysis. And we can talk about that later on in the course of the presentation. Now, when we do all this, there is the need to implement research outcomes, and this is where Ari comes in. We have, Ari will take care of disseminating the results, and one of it is this presentation we are doing here, and I think it's been going very, very well. Now, I will hand over to Jim to continue from this slide.
All right, thank you, George. So George and I are going to tag team this thing here. So I'm going to talk a little bit about the materials and methods and get a start on the results, and then turn it back over to George after a few slides. So the first objective is to characterize the variation and identify genetic markers for FODMAPs and ATI activities in these different wheat lines. Next slide, please. So the panel we use, we assembled 200 different lines. These are ancient, heritage, and modern wheat varieties, and we grew them at both Crookston and St. Paul in 2019. This panel represented a large diversity of our program. So founding breeding lines going back to the early 1900s, and the modern ones I sort of defined as post-1970, which was when the first semi-dwarf varieties were introduced into our region. We treated the plots with fungicide as necessary to reduce damage from fungal pathogens. So we wanted to try to produce the best quality grain as possible. So here's the distribution of the lines that are part of this panel. So these heritage wheats, they were developed prior to 1970. Some of them go back to the late 1800s, kind of the founder line, some of the first wheat varieties that were grown in the regions, things like Fife, Thatcher, which was very common in the 1930s, 1940s. Modern wheats, that's where the main focus was. 142 of those lines, because we wanted to really characterize how much variation for these anti-nutrient traits are present in our current breeding materials, with the thought being that if we see differences there, we can breed for lower levels of these anti-nutrients. The last four, the durum, einkorn, emmer, and synthetic hexaploids, these were more as sort of outgroups to see, you know, what the range of these anti-nutrient activities were in those groups of germplasm. So these are sort of, you know, think of them as relatives of common wheat. Durum is what we use to make pasta. Einkorn is a progenitor of wheat. Emmer is a progenitor of durum. and synthetic hexaploids are made by crossing emmers and einkorns together. So there's kind of a lot of neat little genetic tricks we can do to make different wheat lines. So we looked at all of these. Obviously, we saw a lot of variation in the field for their performance. So we knew we had genetic diversity in our panel. Next slide, please, George. So the FODMAPs were determined using high performance anion exchange chromatography. ATI was done using the HPLC. And then we used a panel of genetic markers to not only characterize the genetic diversity of the material, but also try to find genetic loci that are responsible for the differences that we see in the FODMAP and ATI activities in these materials. So we did a process called genotyping by sequencing to get the DNA markers, which is essentially kind of a modern DNA sequencing technology applied here. It's fairly low cost. It ends up it's basically a genetic fingerprint of each line that we're looking at, and then we use a technique called association mapping to try to find identify the DNA markers associated with both FODMAPs and ATIs. And in objective 2, we won't really hear about this today because this is in process right now, but this is to explore the use of fermentation as a technique to reduce FODMAPs and ATI activity in food products. So we're kind of tackling this from two sides. One is seeing what sort of variation is in the wheat material that is out there, and then trying to tackle it by fermentation to reduce the amount of ATI and FODMAPs in the final products. So the methods for the fermentation, sourdough is going to be prepared from 60 of these 200, and these 60 were selected. So we had a set of 10 that were high in FODMAPs, medium, and low, and then 10 other varieties or lines that were high in ATI, medium, and low. And George will present a list of these at the end. Again, we don't have any results on the fermentation work. I believe the fermentation work has actually been done, but now it's time to characterize the amount of FODMAP and ATI in those final products. So here's some details on the methodology for the sourdough. Sourdough breads are being prepared on traditional methods using a type 1 sourdough culture. All of this is being done by Brian LaPlante with Backwind Foods. The doughs are fermented at 72 degrees at 4, 8, or 12 hours to mimic commercial sourdough bulk fermentation. And then the doughs are being sent to George, and he'll determine the FODMAP and ATI levels of those. So here's our preliminary results. Again, we saw a lot of variation in the field when we grew this entire panel out in 2019. And then we grew a subset of them also in 2020 at both Crookston and St. Paul. So first, the genetic diversity. So this is really kind of a genetic fingerprint type of analysis. So this is a scatter plot, and you can't really see individual lines here, but there are 220 dots, each representing a different wheat line. And the X and Y axes are basically just a means of separating them. So the farther apart any two dots are on this graph, the more genetically divergent they are. So all of the bread wheats are circled in black, and they roughly cluster together. And then these durum, synthetic hexaploids, and emmers are really sort of outgroups as we were expecting, and they kind of form their own cluster. We did have the einkorns in here because our sequencing method really didn't perform very well on this. And if we go to the next slide, this is the same type of analysis, but now we've removed those durums, emmers, and synthetic hexaploid. This allows us to get better separation of the bread wheat lines, and these are the ones that are we're mostly interested in. And again, it's really too small for for any of us to see the names of individual varieties. But as I scanned these, I roughly characterized, these ones circled in blue as the modern lines, so kind of post 1970. The lower right circled in orange as kind of the heritage or founder lines. And then the upper right, black, these are other introductions into our programs over the years, and they consist of Chinese lines. So this is when we were breeding for, well, we still are breeding for scab resistance, but most of that material came from China. We've used Simmet germplasm and South American germplasm to enhance disease resistance or yield over the years, and those are depicted in that section. So we got a really good separation, so we feel like we've got a pretty good level of genetic diversity within this panel.
All right, so I'll take over from here. And so this slide shows how we actually determine the FODMAPs. These are soluble sugars that are easily dissolvable in water. So basically what we did was we weighed a certain amount of the sample. We added warm water to about 80 degrees just to make sure all the FODMAPs would dissolve in there. We dissolved it for about 5 to 10 minutes, filtered it, and ran it on the high performance anionic exchange chromatographic system. So this is what we see when we run the samples. So you can see that you have all the sugars in there, glucose, fructose, sucrose, and all the sugars in there. And then you have the fructans, DP 3, 4, 5, and then 6 to 10. Now, how did we identify these sugars? We actually had to buy standards. So we bought sucrose standards, maltose standards, glucose, and we ran them on the HPAEC system. So we know where they elute. That is how we were able to identify them. Then after that, we developed standard curves for each of these sugars, right? So we prepared different concentrations of the sugars, ran them. So we are able to actually quantify them because that is what we were interested in. So after quantification, this is kind of the results we got. So what we also did was to run, so for each location, we had 2 reps. So for Crookston, we had 2 reps. For St. Paul, we had 2 reps. We wanted to see whether there were differences in the reps in addition to differences in the location itself. So if you look at FODMAPs here, so you can see what we have. We have FODMAPs excess fructose, and then we have FODMAPs free fructose. What is the difference between them? Now, for all these FODMAPs, what is actually the sugar that is actually of primary importance is the amount of fructose. So the body is able to use glucose, so which is okay, but fructose is not easily used up by the body. So therefore they stay in the small intestines, and that is where they cause a lot of irritability, and fructose can be fermented by microorganisms, providing a result in the production of gas. So we are interested in fructose. Now, the excess fructose here was when we subtracted the amount of glucose we have in the sample from the amount of fructose we have. Because we know from literature that if you have the amount of fructose that is utilized by the body is dependent on how much glucose you also have, right? Now, when we did this, we saw that there were some variation in the FODMAP levels in the samples we analyzed for all the locations and for all the reps. You can see that on average, we have about 1% for most of them. And it's ranging from about 0.1 to about 1.5%. For the free FODMAPs with free fructose, it's about 2%. The rep from St. Paul was a little lower, but we had a lot of variation in the rep and one of the rep from St. Paul as well. And then we also have fructans. These are fructans. These are DPs of, I think, 5 to 10. And then we have inulins. Inulins are actually considered as a source of dietary fiber. But if we focus on the fructans with excess fructose, you would think the levels are really low. Yes, 1%. What is really not a big deal. It is actually a big deal, because if you remember, I did say that we have a cutoff point of 0.03% or 0.03 grams per 100 gram. Now, that 0.03 gram per 100 gram should come from the typical serving of food. If we take bread, for instance, this 0.03 grams was based on an approximate of 2 slices of bread. So if you eat 2 slices of bread, you are likely to hit your 0.03 gram per 100 gram if we look at these values here. So yes, the same, if you look at the face value, the same low 1 gram. But if you look at how much we actually need to cause any effect, yes, they still are a lot. And that is why you see these products here having a lot more, having FODMAPs levels beyond the 0.3 grams. So if you should ask me, I would say yes, probably we are, with the levels we are seeing, most of the wheat-based product that we have on the market would actually have FODMAP levels higher than the 0.03 grams that has been speculated to be too much. Now, this slide here actually shows correlations between the parameters that we actually did. Now, this actually would help us to estimate what levels we are getting from the other parameters. So for instance, if you look at FODMAP's free fructose, FODMAP free fructose is positively correlated with FODMAP excess fructose to about 0.82, right? So it means that we should conveniently be able to estimate how much of this FODMAP excess fructose we have if we know the FODMAP free fructose. And this goes on, look, if you look at all the parameters, the correlations vary. Here we see that the fructose is actually negatively correlated to FODMAP's free fructose, right? So, I mean, I don't want to go too much into detail here, but this actually helps us to have an idea as to what levels we are dealing with depending on what factors that we are looking at. I would hand over to Jim again for this slide.
Thanks, George. So this is the results of our preliminary association mapping. So what we're seeing here, this is called a Manhattan plot because it kind of looks like a city skyline with these peaks going up and down. So along the x-axis, we have 21 chromosomes, and each of these chromosomes has probably anywhere between 100 and 200 genetic markers that are kind of laid out in the order that they are on the chromosome. And the y-axis is the strength of the association of each of those 4,000 or so DNA markers with the FODMAP level. So most of these are below this threshold of 1, means it's a very low probability of association. Once we get above about a 3, that's where we have this horizontal line going across. That's kind of our statistical threshold where we would say, hey, it looks like there's maybe something there on that chromosome, a gene that's influencing FODMAP content. So that causes us to focus on what's labeled here as chromosome 14, that blue dot that's above the line, and a couple over on chromosome 20. So this isn't, what we were hoping to see was, all of these levels kind of flattened a little bit lower where they would all be around 1, and then maybe just 1 or 2 really high peaks, like going up to like 5 or 6 on this scale instead of 3. So as a geneticist, what this tells me is that there's no single gene having a major effect on this trait. What this tells me is it's probably a quantitative trait. It's caused by a lot of genes, each with small effect. What that means is, from a breeding perspective, we can probably affect some change, but it's going to be slower and it's going to take a longer period of time, versus if it were a single gene of major effect, we could make changes very quickly in our germplasm. So this is still preliminary, but that's what it's looking like at this point. So to summarize what we've shown on the preliminary findings, we have a genetically very diverse set. We have wide differences in these FODMAPs. We've got no identifiable pattern also regarding the FODMAPs and total fructan versus year of release. In other words, I can't really go back and look at these founder lines or these heritage wheats and say, oh, these tend to be higher or lower in FODMAPs. Same with the modern line. So that's something that we were hoping to see some sort of evidence there that maybe over time breeders have selected for lower or higher FODMAP or ATI activities. And at this point, we're not seeing it. And the last point is that no genomic region is responsible for a large portion of the genetic variation. So it's probably going to be the process of manipulating a lot of genes. And we can do that in our selection if that's the direction that we want to go.
All right, thank you very much. I'll take over now. So the next couple of slides is some results, some data we have on the ATI and also on the fermentation study. So I did mention earlier on that the ATI, we did use the HPLC. And so the first problem we had to tackle was, how does the ATI look like? So we had to buy ATI from Sigma Aldrich. So luckily they had it. So they had extracted and purified ATI that we bought and ran on the HPLC. Now this blue curve here that you see is the ATI that we got from Sigma Aldrich. And then this green line here is the ATI graph for one of the wheat samples that we actually worked on. So from this here, we were able to know that this whole region here is what is the ATI. And therefore, when we are looking at our sample, we have to look at this whole region here. So based on that, we were able to, and based on literature, we were able to divide our graph into 4 major regions. Now, there was a study where they looked at the ATIs where they took these 4 different regions. So this is protein, right? This is all protein. They took these 4 different regions of the ATI protein and then analyzed it further with with an LCMS. And they were able to conclude that the first 2 regions, so this region and that region, contain concentrated amounts of the ATI we are looking for. The 3rd and the 4th regions did not really, they contain very small amount. So for our calculation, we were interested in the areas under the curve for these 2 regions. So this is what we use to quantify our ATIs. All that has been done. And this slide here is just giving you a summary of the ranges that we got. So for all the samples from Crookston, we had, and this is percent, right? 0.007% to 4.7% of ATI. Again, they might seem very small, but when we are looking at proteins, we are looking at how proteins affect our immune system or how they impact our intestinal walls. These levels can still be high. Now, what we found was that the ATI levels in the samples from St. Paul were higher than the ATI levels in the samples from Crookston. So this definitely tells us that depending on where the wheat is grown, you are going to have different levels of ATI. I have no idea why those in St. Paul are higher. Maybe Jim can tell me why. Maybe there are differences in the soil or in the environment, but it was clear that samples from St. Paul had more ATIs than samples from Crookston.
So we actually have got a couple of questions from the audience actually directly related to this.
Okay.
And just because we do have a pretty wide range of people from a lot of different places. Crookston and St. Paul are roughly 250, 300 miles apart, so 400 to 450 kilometers, correct? Mm-hmm. So is there a difference in the environment that might impact the FODMAP and the results of your study?
I guess maybe Jim would have the better answer to this. Jim?
There's definitely big difference in the environments. So where these are grown in St. Paul, it's adjacent to campus. So we're in the heart of the 7-county metro area, and we have much higher nighttime temperatures as well as daytime temperatures than would be experienced during the growing season up in Crookston. So that's a big difference. The soil is quite different as well. Crookston is going to be a much heavier soil, but we're fertilizing them, for a similar level of performance and protein content. Shooting for about a 70 bushel yield goal. So none of these should be short of nitrogen. So the potential is definitely there. I think what we're seeing in the results is, yes, it looks like there are some differences in overall levels, but the variation within any one location is probably quite significant as well. So I don't think we can make a general statement on the variability among these two locations with this limited data. One thing I'll add, and maybe George can expand on this a little bit, is that we have not yet quantified the amount of total grain protein in these samples, and that's something that George is exploring now, because we know that the total amount of ATI is correlated with the total amount of grain protein as well.
Yeah, and just to add to what James said, yeah, even though we have those differences, you probably will see they may not be significantly different. So they may seem higher, but not significantly different. So that is something you might have to have at the back of your head. But yeah, we saw that in here. I probably, so ATIs are considered anti-nutritive factors, and some of these ATIs are produced when the plant is under stress. For some reason, I don't know whether the environment in St. Paul, and probably Jim can talk more about that, is much more stressful to the plants. But yeah, this is what we are seeing. This result might change if we express the ATIs as a percentage of the protein content. These results are as a percentage of the weight of the flour, of the total flour. So we are in the process of determining the protein content of the flour samples to actually express ATI as a percentage of protein.
Okay. Dr. Anderson, has there been any study? I mean, you talked about fertilizer and fungicide. Has there been any evidence as far as crop management impacts on FODMAPs or ATIs?
I'm aware of. I don't know if Georgie, if you're aware of any. Everything that I've seen is looking at genetic differences and not really looking at management practices.
Okay.
Yeah, I haven't seen any study to that effect either.
Dr. Anor, a couple questions around nutrition. So aren't fruit cans good as prebiotics?
So they can be good as prebiotics, but it depends also on so generally people who have irritable bowel syndrome are the people who are more likely to react negatively to these fructans. But generally, yes, they are good. Like inulins, as I said, are actually good for us. But people who have irritable bowel syndrome are likely to negatively react to this, especially when they go beyond that 0.03%.
When you were doing your FODMAP values, were they on the raw material, on the dry pasta, the cooked pasta? What did you run those FODMAPs on?
So these FODMAPs results we are reporting were on the flour. We had the grains. They were, of course, dried grains, and then we milled them, and then we determined the ATIs on the FODMAPs and the ATIs. So
very good. If there's additional questions, make sure you use the chat functions and let us know.
Okay, all right. So we'll move ahead with the fermentation study. As James said, the fermentation study is ongoing. So basically what we did was to select 60 samples, basically looking at high, medium, and low ATI, and I'm going to go through some of the varieties. So Jim and I, and of course the team looked at this. So for the high ATIs, these are the varieties that has been selected. Probably Jim can talk a lot more about these varieties. I'll just go through the list of the varieties that we selected. So these were high ATIs, and then we have medium ATIs, are these samples. And then we have the low ATIs, right? So these are what we selected. These samples are going to be taken through the preparation of sourdough, and then we would reanalyze them to look at how the sourdough process is actually hopefully reducing the ATIs. In addition to the ATIs, we also did for FODMAPs. So these are the samples that we selected for high FODMAP. And then we have medium FODMAPs. These are the samples we selected. So you can see Glen in there. I know Glen is a very popular variety. And then we have the low FODMAPs. So you have the Emma variety here too. So these samples were sent to Brian, and my understanding is that all the fermentation has been done at the different times. Now I'm expecting to receive the samples very soon. And then when we receive the samples, we are going to reanalyze these samples for FODMAPs to see. Now, my anticipation is that the FODMAPs will be much more impacted by the fermentation because the FODMAPs are fermentable sugars, so those would be reduced, hopefully. The ATI, I'm not so sure, but we'll wait and see what would happen after the analysis are done on these fermentation samples. Now, we would like to thank the Minnesota Department of Agriculture for funding this work. We are very, very grateful. Without their support, this work wouldn't have been able to be done. And then, of course, we acknowledge all the team that has been working on this project. My student, Prince, who has worked tirelessly on analyzing. If my memory serves me right, we had to run the ATIs and the FODMAPs on over 800 samples. That was a lot of work. He was in the lab all the time. So yeah, we'll end here with any Q&A. I know there are a lot of Q&A, but yeah, we can dive into the Q&A now. So thank you very much.
Well, thank you, Dr. Anderson and Dr. Honor. So it's an interesting topic, and I think it's one that I know even among family and friends, I got the question last night, right, about within the last 10 years, you're hearing more and more about issues around gluten sensitivity, and for those of us in the wheat industry, it's important to address that. Couple of questions. When will the results of the fermentation study be available, and when will we know the results, and when will they be available?
Okay, so I guess I will take that. So we hopefully should be finishing the analysis in a couple of weeks, probably by the end of next month. We are waiting to get the samples, and when we get the samples, it's all out to get the analysis done. It is not a lot of samples as compared to what we've already analyzed. So my guess is by the end of next month, we should get all the data ready. And as to the release, as soon as we get the data, of course, we'll work in collaboration with RE to get the data out as soon as possible.
What's the next step, right? As we try to figure out kind of some of the underlying issues, what do you guys see as kind of the next steps in this process?
So we've had a couple of discussions on this, and we hopefully are looking at selecting samples that have low ATIs. And when we are low ATIs and FODMAPs, and when we are able to do that, the next step would be kind of to see whether we can increase the amylose content in these wheat samples. Because ultimately we want to increase the health of consumers, and we know high amylose samples are samples that have a lot more resistant starch. So you have a lot more probiotics in there. So that is what we are, the next possible step. And we actually tried to get funding for that, but we're not successful. So now that we are almost done with this study, we are going to try again next year to get that.
Very good. So when you're doing that sourdough analysis, the fermentation analysis, did you compare sourdough to regular commercial baker's yeast?
Brian, you want to comment on that?
I'm not sure if Brian.
Oh, I thought he was in there. Yeah, so we decided to use the sourdough but we are going to compare the sourdough bread. Sorry, we are just looking at the sourdough bread and type 1 sourdough and want to see whether it will result in any reduction in that same sample. We are not interested in comparing it to any other type of yeast. So sourdough bread, if we prepare sourdough bread from these samples, would we be able to reduce the FODMAPs? We hope that if that works, it probably will be applicable in all the other types of yeasted bread.
Is there any current processing technologies that can reduce that FODMAPs or the fructans?
Not that I know of. And not that I know of. And that is why we are looking at fermentation as one of them. The regular bread processing involves some level of fermentation as well. It is not as extensive as sourdough. So if we are able to reduce the FODMAPs with sourdough, my guess will be that regular bread production might result in some amount of reduction, but we are yet to figure that out.
So the next question is, actually, let's, what type of sourdough will you be using?
The type 1.
Type 1 sourdough, very good. There is so unfortunately, Brian LaPlante, who's doing this, is I'm not sure if Grant can get him pulled in as a panelist, but Brian did respond back. Some initial research suggests it takes a very low pH of 3.9 to affect the ATI.
Mm-hmm.
Jim, there's a question directed at you, and I'll start off by saying I'm an economist. I am not a food scientist or a plant scientist. So the question, Jim, in the genetic association study, there are a few points above or close to 3 in chromosome 1, 8, and 14. Are these chromosomes 1A, 1B, and 1D? And if so, do you think there's a major QTL affecting FODMAPs in that region?
So I believe these are the order of the chromosomes from 1 to 21 would be 1A, 1B, 1D, 2A, 2A, 2B. So the short answer to that question is no. Those high points are not just on group 1 chromosomes, but I wouldn't say that any of them are really excited, or I'm not too excited about any one of those particular points because they're just barely at that significance level. What I'm a little bit more interested in is the fermentation samples that we sent to Brian LaPlante were actually from our 2020 harvest, and we sent George grain of those also. So he's going to be doing FODMAPs and ATI on that grain as well. Then we'll have a second year, and it'll be a much more robust type of analysis then, and we'll redo that, and also look and see if we have some genetic regions associated with the ATIs as well.
Mm-hmm.
Brian, thanks for joining the panel. So, and thank you, Grant, for getting Brian pulled up for us. So Brian, I know there's a question. I don't know if you wanted to address. You answered in the chat, but if you had any other comments.
Well, so yeah, we're using type 1 sourdough, which is the traditional ancient method of using a stable culture that is going to incorporate a lot of different bacteria and yeasts as a symbiotic relationship, rather than other approaches that might utilize just 1 or 2 strains of bacteria and or yeast. Modern bread is going to be made out of just a yeast process, but the original yeast that's used in modern processes today was isolated from sourdough cultures well over 100 years ago. So hopefully there will be some overlap in what you would see anyhow from modern techniques to the type 1 technologies. But one of the things that's going to be interesting to see is some of the work that Dr. Genzel did up in Alberta would suggest that the ATIs do take an extended period of time under very low pH to be degraded. So that suggests that traditional sourdough approaches using a longer fermentation and usually achieve much lower pHs could potentially affect the ATIs, but that'll remain to be seen on what we're doing. But we're hopeful that the FODMAPs and ATIs on a longer ferment cycle might be affected.
Very good. Are FODMAPs or ATI located in any particular part of the wheat kernel?
Okay, so not that I know of, but we know that the ATIs, FODMAPs are, of course, carbohydrates, which is more concentrated in the endosperm. And actually, I have a student who was looking at ATI of refined and whole intermediate wheat grass. And we saw that when you do refine the intermediate wheat grass, you get a lot more FODMAPs because you are concentrating the FODMAPs in the endosperm. For the ATI is part of the protein. So anything that results in a reduction in the protein will result in a reduction in the ATI. But I haven't seen any study that has looked at separating the different components of wheat and looking at the ATIs. Yeah.
So you guys looked at a lot of different varieties of wheat, as well as durum and emmer and einkorn. So the question is, would you expect to see any differences with either a hard red winter or a soft red winter type wheat when it comes to FODMAPs or ATI?
Yeah, I would say yes. And we have to dig a little deep into the data we have to look at specific varieties, but our data shows there was a wide range in the levels of the ATIs and the FODMAPs. So my guess is if we go into classify these the wheat into the different varieties, we probably should see differences in the varieties. Yeah, unless Jim want to add anything to it.
Others have looked at this and they do see some differences in winter wheats as well, probably along the same lines as what we're seeing in the spring wheats.
But the difference between the classes, would you expect there to be a stark difference between? Are you saying between varieties or between classes? I guess is the question.
Was saying between varieties, between classes, I really don't know. It would, it could be a little bit confounded by environment where these are grown. Most of the studies they're not comparing both spring and winter wheat grown at the same locations in the same year, so it can be a little bit of an apples and oranges comparison.
So a question coming out around the einkorn and the emmer variety. So you did not take the einkorn all the way through to fermentate or to final product analysis, correct?
We haven't done that so far. I'm not sure whether it was part of the samples we sent to Brian for fermentation, but so far from my side, we've just analyzed the flour. Yeah, and we are waiting to analyze any of the sourdough products sourdough that will be coming from Brian.
So on the emmer and the einkorn, did you see a significant difference on the flour when it comes to ATI and FODMAPs? I haven't dug very deep into that yet. So far, we've just looked at all the varieties as a whole. So as I said, we probably have to go deep into looking at the different variety or classes, as you may put it, to look at whether there are differences between them. But my guess is there should be some differences there. Very good. So that concludes the questions we have. Did you guys have any closing thoughts?
Oh, there's another question here. Does storage time for the grains affect FODMAPs or ATI? We haven't done that. And storage time and ATI, I'm not sure how it would change the ATI and then the FODMAPs. I mean, if the grains are stored the way they have to be stored. So unless there is something that would really, if the grains are not stored well, and of course you have enzymes being activated, then of course they would impact the ATIs. But with the way we store grains, if they are stored very well under dry conditions, I'm not sure what would cause any change. Even if there's any change, I don't think it will be significant.
Very good. Any closing thoughts from our panelists before we let you go?
Well, I would like to say we are very happy to Minnesota Department of Agric. We are very happy to Auri, Backward Foods, Brian, and all the partners in this project. We are happy. We appreciate the Northern Crops Institute for giving us this opportunity to share our data. we still have some work to do. And when all the work is completed, we'll be very happy to share everything out through this medium as well.
Jim or Brian?
I'd echo George's, thanks to the sponsors. And I would say, from a breeding perspective, this is really our first look at it. I view this as sort of a pilot study. And, based on the questions that we got about the environmental effect, I think that's something that we could get at quite quickly. Now that we know, maybe what the varietal variation is within these two locations, we could pick a few varieties. We grow these at 15 locations every year as part of our routine breeding program. We could grab 10 of these from 10 locations, send them over to George if he's willing to do a few more samples, and pretty quickly see what sort of environmental variation is we're seeing with FODMAPs and ATIs.
Sure, I think that would be good.
Very good. Brian, you were a late addition, but any closing thoughts for us?
Well, as you dig into the stuff farther, you find more and more complexity, and you find that over time, how foods are processed plays a major role in the finished good and how it is for consumers. And I think these are initial steps, and they're important initial steps to unraveling some of these mysteries, because this is a thing that's developed over a long period of time, and there's a lot to be learned in understanding the human microbiome and how food reacts with it. So there's a lot of things that come into play.
Well, thank you for joining us today. Thank you for the excellent presentation, and thank you for all the good work that you do. So very interesting results, and we will take you up on your offer when you get the next round of results. We would
Sure.
welcome.
Sure. Yep.
Thank you.
All right. Thank you.